Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speaker, Bo Jiguan, PhD, Senior Scientist, Department of Viral Vector Process and Technology Development for WuXi Advanced Therapies.
Thank you for coming to the webinar. Today, I'll briefly introduce the Streamlined service of AAV manufacturing our company, WuXi Advanced Therapies, is offering from Plasmid design to GMP production. I'm one of the team leaders in the Viral Vector Process Technology Department. WuXi ATU is an international company that provides contract testing, development, and manufacturing service to cell and gene therapies. We offer a streamlined service from as early as plasmid construction, R&D materials preparation, different phases of clinical trials, up to the commercial production and the biologics license applications. Our viral vectors include, but not restricted to, adeno-associated virus, lentivirus, herpes simplex virus, and adenovirus. Our cell therapies cover CAR-T treatment, tumor-infiltrating lymphocyte, and stem cell-based therapies. We also have an independent and integrated testing group that is dedicated to the standard QC assays for IND filing. We have facilities across 3 continents designated for specific functions. The U.S. Philadelphia campus has 30 GMP suites and does most of the process development, manufacturing, and testing for cell and gene therapies. The UK Oxford site came on board after acquiring Oxygen in 2021, and it contains state-of-the-art facilities for most research activities, including plasmid design, construction, and optimization. Also cell line development, such as the newest AAV platform TESA technology and the lenti-stable producer cells. The China, Shanghai, and the WuXi City are in charge of all the plasmid manufacturing for both the GMP and R&D grade quality. It also covers some of the AAV and the lenti process development and the manufacture overseas. This is a brief map of how we coordinate our services geographically and functionally in supporting the different phases of drug life cycle. Overall, our main goal is to help our clients in delivering their breakthrough therapies to the patients who need them. WuXi ATU started AAV manufacturing since 2015 with collaboration with company Regenx Bio and the University of Pennsylvania. The first 5 years of our AAV production is based on adherent cell culture system. Since 2019, we have fully transitioned to serum-free suspension system. With continuous improvement and single clone cell line development, we have established 2 current AAV manufacturing platforms. One is transient plasmid transfecting system, AAVX suspension. The other is next generation inducible producer cell line, TESA AAV. For the purpose of this webinar, we will focus on the AAVX suspension platform. The AAVX platform uses triple plasmid co-transfection of gene of interest, serotype-specific wrap cap, and the universal helper to produce 4 capsid AAV from the upstream HEC-293 suspension cells, followed by several established lines of downstream purification. We aim to provide all components, processes, and testing requirements to support clients through to clinical development. We have dedicated GMP suite in both the US and China that can provide the production up to 200-liter scales, along with process and analytical development and supporting of tech transfer and the vector-specific assays. One of the unique features of WuXi AAVX is our patent Plasmid technology derived from oxygen designs and constructs that improve AAV vector productivity and quality. Reconfiguration of the red cap plasmid cassette allows a well-balanced expression from each gene for maximum AAV production. The significant improvement of yields has been validated in at least AAV serotypes 2, 5, 6, 8, and 9 at different scales. Other than the plasmid quality, through a series of single clone cell cloning, transfection, and evaluation, WuXi AAVX selected the optimal suspension HEC293 clonal cell line with robust cell growth and viability, low cell aggregation, and remain stable after multiple passages. Our optimal cell line has shown to increase 2 to 3-fold of AAV titer depending on the serotypes. Most importantly, our master cell bank and working cell bank are fully banked and release tested, ready to go for any of client's projects that guarantee a significant time saving. The way we establish our serum-free suspension system is by gradually adapting our initial adherent HEC293 cells to chemically defined media, and eventually using the selected optimal clones for process development and manufacturing. We are confident that our research cell bank, master cell bank, and working cell bank all behave similarly with robust growth, phenotype, and consistent high titer of AAV production. WuXi offers a variety of upstream scales depending on the purpose of the production, from shaker flask of plasmid evaluation, smaller bioreactor R&D material preparation, 10-liter and 50-liter of feasibility or verification runs, up to 50-liter and 200 liters of pilot engineering and GMP runs. We have consistently delivered the high productivity of almost all serotypes of AAV in smaller to mid-sized bioreactor scales and demonstrated some selected serotypes in larger scales. This upstream workflow is the recipe of how we do it stepwise, from cell inoculation and expansion, cell seeding and transfection, to the eventual cell lysis and harvest. The key factor to this recipe, such as cell density, transfection reagent, plasmid ratio and concentration, days of harvest and cell lysis condition, have been all sorted out and integrated into our platform service. This downstream purification workflow summarizes the 3 platform options AAVX offers, depending on the client's preference of final drug product purity and the overall process recovery. The purification recipe starts uniformly with clarification and the AAV capturing chromatography. After that, in order to separate full capsid AAV from empty and partially packaged AAV, you can either go for another AEX chromatography, we call it 2.0, or using ultracentrifugation 2.1, or you can choose to combine both 2.2. In practical, this would need some process development to narrow down the best condition for the gene of interest and the AAV serotype of client's interest. The last polishing step is the final gentle touch to exchange buffer with final formulation, concentrate, sterilization, and to have drug products filled into proper vials and volumes. In reality, as more complex the processes are, the eventual purity is higher, up to almost 90%, but comes with the cost of lower recovery, around 20%-ish. Overall, the purity around 80 to 90% are quite impressive from the standpoint of in vivo gene therapy application. In conclusion, we believe WuXi AAVX offers a streamlined AAV manufacturing service integrated with standardized upstream and downstream platforms to enable a quicker turnaround for early clinical phase application of cell and gene therapies. To summarize, with our patent Plasmid design, high-performing cell line, and standardized upstream and downstream processes, the WuXi AAVX suspension is not just an optimized AAV manufacturing streamline that are easily scalable, efficient, and consistent. It gives a certain flexibility for client tailored needs in production to guarantee the highest likelihood of project success and delivery. If you are interested and have more questions regarding our service, please reach out to WuXi Advanced Therapies, and I thank you for your time and attention.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what is your plasmids ratio for AAV production?
So the plasmid ratio is solely depending on the serotypes. So different types of AAV comes with different ratios. Some of them use equal ratio of 1 to 1 to 1, but some of them use a higher ratio of the GOI, depends on the percentage of the full capsid. This also rely on the client's request for the small scale DOE that is needed before we go up to the larger scale.
And how do you decide which downstream platform process to go per client?
Again, this is very tailored for client's request. So depending on the client's demands, a higher purity or higher recovery or the overall eventual yields. And also this depending on the application for in vitro or in vivo. For example, if this viral vector is designed for in vitro, it usually doesn't need that highest purity, and so you can choose like 2.0 platform that has a higher recovery, but with a little bit lower purity, around 78 to 80%. But if this is for the in vivo that is going straight to the patient, then we would prefer to go the highest process to get the highest purity possible. And also this depending on the clinical phase. So different platform has different challenge to go scale up to the GMP scale. So you can tell if ultracentrifugation is involved, then the scalability is a challenge, but we can work around it depending on the client's request.
Which AAV serotype in your experience is more challenging than others? How do you overcome the hurdle?
That's a good question. So from my personal experience, AAV2 is more challenging than the others. It's well known that the AAV2 is usually have a lower yield and very easy to aggregate. So for WuXi process development department to overcome this hurdle is we have our own dynamic light scattering machine to monitor each steps of the cell viral vector aggregation. So we also try different formulation buffers and pH adjustment, try to not get the AAV to the over concentrate titer that leads to the aggregation. So with all this assay on the side, we can very carefully monitor each of the steps to overcome the hurdle.
How consistent is your cell line for AAV production over the cell passage number?
From our own process development data, we can guarantee from P4 to up to P20, the cell line is very consistent and stable with high AAV production.
Okay, it looks like we're running short on time, so if we don't get to your question, we'll be passing them along to Bo, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how robust is your platform? Can it fit to all serotypes?
So this is regarding to the downstream process. So for the clarification and the capture chromatography is uniform for all serotypes. So we use the same depth filter and the column for the capturing. And then after that, the AES chromatography will need some scouting. So this will depending on the process development case by case. And the final ultracentrifugation, again, is depending on the final drop product is going for the in vivo or in vitro, and the different early phase or commercial production. So this the platform is we have worked on different AAV serotypes, so we can assure the 3 options of the downstream is guaranteed to fit all serotypes.
Okay, well, thank you, Bo.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be made available for viewing on demand on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.